Hi students, this is Aisha, a third year MBBS student. Today in this video, let us discuss about excretory products and their elimination. How excretory products are formed and how they are eliminated from the body in a unit called human physiology. Okay. So, let us discuss the introduction of excretory products and their elimination. So, what are the major excretory products in our body? Those are nitrogenous waste. This nitrogenous waste need to be eliminated through the body. Okay. Those nitrogenous waste are ammonia, urea and uric acid. This ammonia is a most toxic nitrogenous waste. Okay, and it requires more amount of water to be eliminated. Okay, that is why it is most toxic, and uric acid is it is least toxic. Why it is least toxic? Less amount of water is required for elimination. It does not require more amount of water, only less amount of water is required. That is why it is least toxic. And urea is a nitrogenous waste which is produced in the mammals, that is human beings. Okay, along with nitrogenous waste, ions like sodium ion, chloride ion, and this is potassium ion, phosphate and sulfate ions are eliminated, and little amount of CO2 is also eliminated. Majorly CO2 is eliminated through gaseous exchange, but through excretion also little bit amount of CO2, and water is excreted. Okay, see, through metabolic activities, that is chemical reactions or excessive ingestion, more eating. Okay, in animals these products will be accumulated, will be stored in the animals. This need to be eliminated either totally or partially. Little amount of nitrogenous waste or these ions or this water should be eliminated or totally need to be eliminated depending upon the toxicity of the body. Okay, toxicity of the content in the body. Okay, see, as I discussed above, same, ammonia is more toxic, require more water for its elimination, and uric acid is least toxic and eliminated with least amount of water. See, the animals which excrete the ammonia, those are called ammonotelism, and the animals which excrete the urea, ureotelism, and the animals which excrete the uric acid, those are uricotelism. Excrete urea, ureo, and excrete uric acid, uricotelism. Okay, we have to remember the examples in this. Okay, now bony fishes are there, no, and I told you ammoniotelism means requires more water. That's why aquatic amphibians and aquatic insects adapt this. Okay, in these animals only ammonia is produced and excreted. How ammonia is soluble in water? There will be lots of water, right? So it is soluble in water and excreted by diffusion through the body surface and gills in the fishes. Okay, through body surface diffusion process only ammonia will be excreted, or gills are there, no, in the fishes. Through gills ammonia is excreted. And ureotelism means which excrete urea. What are the animals? Mammals. I told you mammals and terrestrial amphibians who live on land. Okay, and marine fishes called chondrichthyes. Okay, what is uricotelism? Excrete uric acid. What are the animals? Reptiles, birds, land snails and insect. Okay, they will excrete uric acid in form of pellets. Pellets will be there, no, small circular hard structures, no water will be present. Pellets or paste like. Okay, in the form of pellets or paste with minimum loss of water, uric acid will be secreted. That is called as uricotelism. Now we told that in mammals urea is produced and it is excreted as a nitrogenous waste, right? Let us see how urea is produced. In liver, you see that amino acid is there? No. Amino acid will break down and will release ammonia in the liver, and ammonia will be converted into urea by ornithine cycle in liver only, and this urea will be sent through blood to kidney. Okay, in the kidney urea is filtered through many processes. Urea will be filtered and excreted out of the kidney. Okay, this is the process of urea production and excretion. Excretion we will discuss in detail in the further slides. Okay, so urea is excreted and some amount of urea is there, no? It will remain in the kidney matrix, in the kidney fluid only. Some amount of urea will be remained there to maintain osmolarity, to maintain the concentration, osmolarity. Urea will be present in the kidney matrix. Let us discuss about the excretory structures. Okay, so main excretory structures, actually this should be covered in animal kingdom, but remember these few excretory structures and the animals in which these excretory structures are present. So protonephridia or flame cell, nephridia, Malpighian tubules and antennal or green gland. Okay, look at their function. Their function is fluid and ionic balance, that is osmoregulation, fluid and ionic balance and removal of nitrogenous waste. What is excretory structure function? Nitrogenous waste excretion, removal of nitrogenous waste and osmoregulation, that is fluid and ionic balance. Excretory function. Okay, so function is same, that is osmoregulation, fluid and ionic balance and removal of nitrogenous waste. So question will asked, what are the animals and what are the excretory structures. So in platyhelminthes like flatworm, example planaria and rotifers, some annelids and cephalochordata like amphioxus, they have protonephridia or flame cell. Okay, you can see in earthworm and other annelids, some annelids will be having protonephridia and other some annelids will be having nephridia along with earthworm. And most of the insect like arthropod, cockroach is present. Cockroach has Malpighian tubule. Okay, and crustaceans like prawns will be having antennal or green gland. Okay, you have to remember the excretory structures and animals which are having those excretory structures. Okay, let us discuss now about human excretory system. What human excretory system? Excretory system is which excrete the nitrogenous waste and other substances through kidney, and this is the pathway. See what excretory system contains: a pair of kidney. See, you can see a pair of kidneys present, 2 in number, and this kidney will lead to ureter. Ureter is a tube. It is also a pair, 2 in number. And then urinary bladder. Urinary bladder in which the urine is stored, that is 1 in number, and this is urethra, that is also 1 in number. Through this urethra urine will be released out. Okay, so this is the, you have to remember, a pair of kidney and a pair of ureter and 1 urinary bladder and 1 urethra. Okay, it is part of excretory system. Now let us discuss about kidney. Kidney, you can see here, it is reddish brown in color, and it is bean shaped. Have you seen beans? Beans will also be in this shape only. So it is reddish brown and bean shaped, and it is situated between last thoracic vertebra and third lumbar vertebra. See, now any organs are present in our body, it will be compared with respect to the vertebral position. Okay, see, last thoracic vertebra they are saying means T12 vertebra, and first lumbar vertebra second lumbar vertebra and third lumbar vertebra. In between these two, kidneys are present. Last thoracic vertebra and third lumbar vertebra, kidney is situated between them. Okay. So next, actually kidneys are situated dorsally, means back side. See, I told you this diaphragm is there, no? Diaphragm will divide into thoracic cavity, above is thoracic and lower is abdominal cavity. Kidneys are located in the abdominal cavity, inner wall of the abdominal cavity, close proximity, where close to dorsal side. Dorsal means back side. Okay. And now let us discuss each kidney. These are the measurements. You can see this is the length. Length is 10 to 12 centimeter and width is 5 to 7 centimeter, and thickness, you see this thickness is there, no? Thickness is 2 to 3 centimeter and weight is 120 to 170 grams. Remember this, okay, measurements of kidney. Now let us see the pathway of the excretory product elimination. Okay, see, kidney is covered by tough capsule that is called as renal capsule, and in kidney, inside the kidney, this is a section, longitudinal section from above of the kidney. In the center they made a cut, so that see, two equal halves will be cut out. Okay, so first half we are taking a longitudinal section, and transverse section means which is cut from here. Okay, longitudinal section, you can see the outer layer, that is cortex, up to this pyramidal structures are there, no? On the top of pyramidal structures and the outer layer, this is cortex. This is cortex. Okay, these pyramidal structures are there, no? This is medulla. Okay, see, what is this then? This is also cortex, which is extending between the medulla. It is called as columns of Bertini. Cortex portion in the medulla, it is columns of Bertini. Okay, now this medullary pyramids will release the fluid or filtrate in the urine and the these are the calyces. Okay, this different calyces from different medullary pyramid will join together into a renal pelvis. Okay, renal pelvis will open outside of the kidney, that is called ureter. Okay, and here you can see this is inside. See, here there is a notch, no? There is a notch like structure here in the center of the kidney. You can see there is a notch which is inner side is concave. See, inner side, this is inner side, this is outer side, no? Inner side is a concave like structure, it is called as hilum. Okay, towards center, inner concave surface of the kidney, that is a notch, that is hilum. Okay, so from ureter, again you know, it will go into the urinary bladder and urethra and excreted out. So once again, here I also made a diagram, let us discuss here. So this is a cortex, that is outermost layer, and outer layer of the kidney by tough capsule, and here medullary pyramids are there, no? This is medulla, that is divided into medullary pyramids. These are conical mass. See, cone like conical mass, medullary pyramids. And in between the medullary pyramids, you can see cortex portion in the medulla, that is columns of Bertini, and this medullary pyramids will open into calyces, and many calyces into renal pelvis, and then finally into the what? Ureter. Okay, see, outer side to inner side. How? Cortex, medullary pyramid, calyces, renal pelvis, ureter, urinary bladder and urethra, and excrete out the urine. So this is the pathway. Now, okay, this is also same thing but little more explanation. See, each kidney is there, no? We have a pair of kidney, but each kidney will be having 1 million nephrons, that is 10 lakh nephrons in each kidney. So both the kidneys will contain 2 million or 20 lakh nephrons. Nephrons are, we know from childhood that it is structural and functional unit of kidney. Okay, these are complex tubular structure. This is only a nephron structure. See students, it is a complex and tubular structure. Tubes are there. Complex tubular structure. Okay, let us discuss about the parts of nephron. Before going ahead, let us discuss about parts of nephron. See, we all know it is made up of each nephron. Each nephron has two parts, that is glomerulus part. Glomerulus means capillary part, and this is renal tubule part. Okay, see how glomerulus is formed. You have to remember this. Blood will be supplying to the kidney, right? How blood will be supplying? Through artery. The artery of kidney is called as renal artery, and renal artery will be broken down into afferent arteriole. Afferent arteriole. In Hindi they will say come. For come we say aja, right? So you have to remember this renal artery is going near this Bowman's capsule. Okay, that is why come means aja, right? That is why afferent arteriole. And this arteriole will be broken down into capillaries, and then it will form, generally what happens, arterioles and capillaries, and then what it forms vein, right? No, here vein is not formed. Here arteriole formation only will occur. Still it is supplying the blood throughout the nephron. Up to here only it will break into capillaries and then it will form vein. No, it's not correct. Here again it has to supply blood in the other parts of the nephron. That is why it will form efferent arteriole. Okay, renal artery, afferent arteriole, glomerulus, that is capillaries, and efferent arteriole. Okay, this glomerulus is there, no? This is a part of nephron. This capillary network is there, no? Tuft of capillaries, that is a part of nephron. And here tubule, renal tubule. What renal tubule contains? This is Bowman's capsule. See, Bowman capsule. It is a double-walled Bowman's capsule, which will lead to PCT, that is proximal convoluted tubule. Proximal means first, beginning, which is attaching with respect to glomerulus. Okay, proximal convoluted tubule. This is loop of Henle. It is a hairpin like. See, hairpin like. And then DCT. DCT means distal. Distal means far. Proximal means near. This is near to glomerulus or Bowman's capsule. It's proximal convoluted tubule. This is distal convoluted tubule. Many DCTs from many nephrons will open into collecting duct. Okay, this collecting duct is not part of nephron. Okay, nephron ends up to here and different nephrons will be there. No, again DCT part, they will all together open into collecting duct. Okay, you have to remember collecting duct is not a part of nephron. Okay, see, Each nephron has glomerulus and renal tubule. Glomerulus is a tuft of capillaries. It is a tuft of capillaries, and Bowman's capsule is double-walled cup-like structure. It is like cup, right? It is a double-walled cup-like structure. And Bowman's capsule will open into PCT. PCT is a highly coiled. You see how much it is coiled? Highly coiled tubular structure. PCT will lead to loop of Henle. Loop of Henle is hairpin like shape, and it has, see, this has descending loop of Henle. It is going downwards, and this is going upwards. Ascending loop of limb of loop of Henle. Loop of Henle has two limbs, that is descending limb of loop of Henle, ascending limb of loop of Henle, which leads to highly coiled DCT. DCT is also highly coiled as PCT. Okay. Now many DCTs of nephron, different nephrons will open into collecting duct. Okay. Now through collecting duct, this urine will be there, no? Will be poured into the medullary pyramid. Through medullary pyramid, okay, what happens? It will be poured into renal calyces, renal pelvis, ureter, urinary bladder, and urethra. Okay, from nephron, in nephron of the kidney, all this filtration process is occurring, and ultimately urine will be collected in the collecting duct, which will open into the renal calyces through medullary pyramid. Renal calyces, then renal pelvis, ureter, urinary bladder, and urethra. Okay, this is the passage which I am saying in nephron. See, this is glomerulus from afferent arteriole. See, one more thing which you have to remember is this afferent arteriole is thicker, wider. Okay, this efferent arteriole is thin, small. Why? Now see, this width is more, no, in the afferent arteriole, so more amount of blood will be passed into glomerulus. Okay, this efferent arteriole is thin, so less amount of blood will be going through the efferent arteriole. Means what? So much blood will be stored here, which increases the pressure. Through pressure, it will be filtered into the Bowman's capsule. That is why naturally afferent arteriole is thicker and this is thinner. So much blood will be going into it and least amount will be coming into efferent arteriole. So here they will be having so much time. Blood will be attained here in the glomerulus for so much time with more pressure so that it will be filtered. Okay. So that's what here I have mentioned. Efferent arteriole is thinner than afferent arteriole. So here this is afferent arteriole. See, this is thicker and glomerulus, and this is efferent arteriole is thinner. Okay, this is Bowman's capsule. See, Bowman's capsule, which lead to PCT, proximal convoluted tubule, and this is loop of Henle. First, this is descending loop, and this is ascending loop of Henle, and this is DCT, coiled structure, and many DCTs will open into collecting duct. And one more thing which you have to remember here is through afferent arteriole will go away, no? This will give rise to one more capillary called peritubular capillary. Okay, this peritubular capillary will form a network of structure, blood structure around the loop of Henle. This is called as vasa recta. Okay. Now loop of Henle is there, no? Loop of Henle will be descending and then ascending and form proximal convoluted tubule, no? Now vasa recta will be there, no? See, I will show you. This is loop of Henle, direction is this, okay? And vasa recta will be there, no? It will form a direction reverse to the loop of Henle. See? You have to remember this thing. Here, I'll just tell you the concept here. This loop of Henle is there, no? it is descending and then ascending. Here in between the two limbs of loop of Henle only, direction is different, no? That is counter. Counter means opposite. Counter current. Current means some mechanism is occurring. So this loop of Henle, two limbs of loop of Henle will be having counter current mechanism. And here also this vasa recta is there, no? Within vasa recta only, see, this is going in downward direction, this is going in upward direction. Here also opposite, counter current mechanism is there. And if you check between loop of Henle and vasa recta, it is moving upwards and it is going downwards. So between the proximity of the loop of Henle and vasa recta also there is counter current mechanism. Okay, in further slides also I will explain, but just have an idea. Loop of Henle direction is this and vasa recta direction is like this, counter current mechanism. So next one is, what you have to remember is Bowman's capsule is there, no? Bowman's capsule and this glomerulus, this network of tuft of capillaries, it will be called as Malpighian body or renal corpuscle. See, Malpighian tubule is an excretory structure in insects, okay, like cockroach. But Malpighian body or renal corpuscle is Bowman's capsule plus glomerulus. So now let us discuss different type of nephrons are present in kidney. Okay, based on the location, different type of nephrons are present. See, we all know this is the cortex region and this is medullary pyramid, means medulla. Here nephrons are usually present in the cortex region. Okay, this is Bowman's capsule, this is PCT, this is loop of Henle, DCT. This loop of Henle is dipping into the medulla only little amount, see, only little. Whereas some nephrons are there, no? This PCT, they will be present in the deeper side of the cortex. This is a loop of Bowman's capsule, this is PCT, and look at the loop of Henle. It is going very deep into the medulla and then coming out as DCT. Okay, so the loop of Henle which is present in deeper side of the medulla is juxtamedullary nephron, and the loop of Henle is touching just little part of the medulla, it is cortical nephron. Cortex, that is why cortical nephron, majority of nephron is present in the cortex, that is why cortical nephron. And juxtamedullary means this loop of Henle is present deeper into the medulla, that's why juxtamedullary nephron, in the name itself medullary. So let us differentiate more. Loop of Henle is dip very little into the medulla, and loop of Henle is short loop of Henle is present. That is why loop of Henle will dip very little into medulla. And see, majorly this Bowman's capsule, PCT and DCT, they will be present in the cortex, and through the blood vessels it will be supplied. Okay, so that's why vasa recta is not required, it is reduced or absent in cortical nephrons, whereas loop of Henle is very long in juxtamedullary nephron and runs very deep into the medulla, and that is why it has to be need to supplied by the blood vessel, no? That is why vasa recta is also present in juxtamedullary nephron. You can see I told you this vasa recta. Here this is a renal artery, afferent arteriole, this is glomerulus, these are capillaries, and this is efferent arteriole. It will broken down into different capillaries, and one peritubular capillary will be formed from efferent arteriole, and peritubular capillary will form a U-shaped tubular structure around the loop of Henle, that is called vasa recta. See the direction of loop of Henle, it is like this, and vasa recta direction is opposite, that is why counter current mechanism. So students, let us discuss about urine formation. This is the most important topic in excretory system. Okay, and if you understand the concept, no, this will become easier. I hope I will make you understand easily. So urine formation means now what happens generally, blood will be there, no, we discussed right here, this is the blood capillary glomerulus. It will be pouring the blood into the Bowman's capsule. Blood will be filtrate. Okay, now whole blood won't pass into the Bowman's capsule, only the filtrate will pass into the Bowman's capsule. This filtrate composition is there, no, that is same as, this filtrate composition is same as plasma. Plasma will be there, no, blood components. What are the blood components? Formed elements and plasma, right? Plasma also have proteins like albumin, globulin and fibrinogen. If you remove these proteins, and the composition of plasma is same as filtrate, which will be passed into the Bowman's capsule, and at last it forms urine. Okay, that's what here we are discussing. In urine formation, first what is the first step? Glomerular filtration. In the glomerulus filtration will occur, which will go into the Bowman's capsule. Second one is reabsorption. So let us discuss in detail further reabsorption and then secretion. Okay, first step is glomerular filtration. What happens in glomerular filtration? See, first step in urine formation is filtration of blood. Okay, blood should be filtered, and that is carried out by glomerulus. This glomerulus tuft of capillaries will filter the blood. Okay, that is called glomerular filtration. See, we discussed in circulatory system, the cardiac output is 5 liters, right? In this, 1/5 of blood will be filtered by kidneys. 1/5 means, you only tell me what will be the amount. 1.1 liter to 1.2 liter. 1/5 means out of 5 liters, 1 liter should be filtered out by the kidney, right? So roughly 1/5 of the blood pumped by each ventricle per minute, that is 1100 ml to 1200 ml of blood is filtered by kidney per minute. Okay, so this is just the explanation. Now each ventricle will be pumping some blood per minute, right? That is 70 ml stroke volume. Okay, stroke volume into heartbeat is cardiac output, and cardiac output will come out as 5 liter. So roughly 1/5 of the blood pumped by each ventricle, that is stroke volume into per minute, that is heartbeat, that is cardiac output. Out of 5 liter, only roughly 1/5 of blood will be pumped through the glomerulus, a tuft of capillaries, into the Bowman's capsule, that is 1100 ml to 1200 ml. This is important, you have to remember the value. So here you can see how the filtration process occurs. In filtration there will be layers, right? Through that layers only filtration process will occur. Here what are the layers of filtration? Okay, glomerular filtration. Here this is glomerulus capillary, right? It has endothelial cells of glomerulus. Endothelial, epithelium of the endothelium. Inside the capillaries, endothelial cells are present. And here in Bowman's capsule, epithelial cells are present. Okay, This endothelial cells of glomerulus and epithelial cells of Bowman's capsule, and in between the glomerulus and Bowman's capsule there is a basement membrane. Okay, through these membranes 1, 2, 3, blood in the afferent arteriole will be flowed into the Bowman's capsule. Okay, and one more important feature which we have to remember in Bowman's capsule is Bowman's capsule will be having this epithelial cells are there, no? They are modified as podocytes. See, podocytes means here like this, podocytes, modified podocytes. Okay, this epithelial cells will be having podocytes. They are arranged in intricate manner. Means this is a podocyte. Okay, and this capillaries will be there, no? Capillaries will be held by the podocytes of Bowman's capsule on either side. On either side it will be held and it will be closing it so that little space will be present to passage of the blood into the Bowman's capsule. See, Bowman's capsule has podocytes. This is one side of the podocyte. Bowman's capsule podocytes will encircle the glomerulus, and in between you just imagine if you keep a pen between the in your palm, these fingers will be enclosing the pen. Okay, in between the fingers there are some gaps, right? Through that gaps are called as filtration slit or slit pores. Through that gaps only blood will be filtered, and the fingers which are encircling the pen are known as podocytes of Bowman's capsule, and the pen which is encircled by the fingers is called as what is that? Endothelial cell of glomerulus. Okay, I think you it made it clear, and you remember podocyte how they will be arranged in the form of intricate manner. Okay, so this is about glomerular filtration. Still it is there, glomerular filtration. See, so roughly how much blood will be filtered by the each kidney per minute? That is 1100 to 1200 ml of blood. That is roughly 1/5 of cardiac output. Okay, see, amount of filtrate. Now blood is there, no? Here blood will be filtering. Okay, only filtrate will enter into the Bowman's capsule. Filtrate is same as plasma except proteins. Okay, blood won't be filtering into the nephron. From blood only filtrate will pass through the Bowman's capsule. Okay, that is same as plasma except proteins. See, amount of filtrate formed by kidney per minute. Okay, how much amount of filtrate is formed means filtration process will occur in the glomerulus, no? That roughly 110 ml to 1100 ml to 1200 ml of blood will be filtered in the kidney, right? Glomerular filtration. How much amount of filtrate will be formed by the kidney per minute? That is called as glomerular filtration rate. And how much healthy glomerular filtration rate in healthy individual? That is 125 ml per minute out of 1100 ml to 1200 ml of blood. Only 125 ml per minute of filtrate is formed in the Bowman's capsule. And per day how much filtrate will be filtered? Then 180 liters. From where 180 liters of GFR will be formed? It is highly impossible. 180 liter of urine will be filtered out? No. Maximum 1.5 liter of urine will be excreted. What about this remaining 180 liter of urine? This will be reabsorbed. Okay, this will be reabsorbed in the tubules, and ultimately 1.5 liter of urine will be excreted. Okay, that is why the second step is reabsorption. So you understand, in an healthy individual 125 ml of filtrate will be filtered per minute, that is 180 liters per day. Okay, kidney have built-in mechanism to regulate the GFR. Now GFR, glomerular filtration rate, should be regulated. Suppose if GFR, glomerular filtration rate, is decreased, means blood flow is less. Okay, blood flow is less because pressure is less, so GFR will decrease. No, this rate will decrease. That is why kidney have their own mechanism to regulate the GFR, that is called as JGA cells. Okay, JGM is juxtaglomerular apparatus will be there, which will regulate the GFR. What is juxtaglomerular apparatus? See, this is Bowman's capsule, this is PCT, this is loop of Henle, this is DCT. In DCT, some specialized cells will be present, and in afferent arteriole, some specialized cells will be there. They will form a complex, afferent arteriole, and in DCT, macula densa, which is not mentioned in NCERT, but macula densa is a specialized structure in DCT, and in afferent arteriole, they will both form a complex, that is called as cellular modification will occur. Afferent arteriole and DCT, afferent arteriole cellular modification, and juxtaglomerular apparatus will be formed. Okay. So if there is any fall in GFR, what will happen? First, this juxtaglomerular apparatus will activate. Okay, JG cells will activate, and this JG cells will release renin. See, RENIN. I already told you, kidney will be having two in number, no? That is why it will be reverse. That is only one N will be there in kidney, whereas stomach is a single organ, right? That is why REIIN, two N's will be produced in the stomach. So it is the way of remembering the renin. Okay, so single N, renin, will be released in the Bowman's capsule by JG cells, and it will stimulate the glomerular blood flow. Blood flow will be stimulated, and pressure will increase, and GFR will increase. So I think I am very clear. This glomerular filtration will be having its own mechanism in regulating the GFR, that is JG apparatus. Okay. See, now reabsorption. I told no, volume of filtrate formed per day is 180 liter per day, but volume of urine released is 1.5 liter. So what is the meaning of this? 99% of filtrate is reabsorbed by renal tubules. Now in Bowman's capsule, the filtrate is formed, no? In the renal tubule, reabsorption will occur, 99% filtrate reabsorption. So that ultimately 1.5 liter of urine will be formed out of 180 liter per day. Okay, now what are the products which are reabsorbed? You have to remember this, that is sodium ion will be reabsorbed, and glucose molecule and amino acid molecule. These two molecule and this ion will be actively reabsorbed. Okay, and nitrogenous waste is there, no? Passively it will be reabsorbed. You might be thinking nitrogenous waste is a waste product, harmful product, it should be eliminated, right? But majority will be eliminated, but least amount will be reabsorbed back into the body. We discussed, some amount of urea will be present in the kidney matrix to maintain the osmolarity. To maintain the osmolarity, this urea, nitrogenous waste, will be passively reabsorbed. Okay, and H2O, water, also will be passively reabsorbed in the initial segment of nephron, means PCT, like that. Okay, so you have to remember the products actively absorbed, and nitrogenous and water passively absorbed. Okay, We are done with the second step, that is reabsorption in the formation of urine, and last step of formation of urine is secretion. Okay, in secretion what happens? What are the ions which are secreted into the renal tubules? From renal tubules, these products were reabsorbed, okay, but some products will be secreting in the renal tubule. What are the ions? What are the products? H plus ion, K plus ion and ammonia. Remember this, mug up it, okay. H plus ion, K plus ion and ammonia, these are secreted into the filtrate, okay. In the filtrate, in the tubules, these will be secreted, okay. Why these are secreted? What is the reason? We know reabsorption, why it is occurring, because this much amount of liters of fluid can't be excreted, right? This much amount of filtrate can't be excreted as urine. Only 1.5 liter is excreting. What about the remaining filtrate? That is reabsorbed. Now what is the function of this secretion? To maintain the ionic and acid balance of the body fluid, okay. To maintain ionic balances, this will be secreted. Now see, sodium will be reabsorbed, no. So positive ion is going away from the tubule. To maintain it, H plus will be secreted in the tubule. That is why maintenance of ionic and acid balance of the body fluid, this H plus, K plus and ammonia is secreted, okay. This is about the urine formation. Now let us discuss in detail about functions of tubules. See, first we will discuss about PCT, proximal convoluted tubule, loop of Henle and DCT and collecting duct. See, very simple. By this we will complete the major topic, okay. If you learn this functions of tubules, no, it is so easy. We will be completed the major topic and remaining left out part is only simple thing, okay. So here first we will discuss about the function of PCT. What happens in PCT? So in PCT, what are the cells? That is simple cuboidal brush border epithelium. Cuboidal cells are present in PCT and they are brush bordered, okay, brush bordered. Why brush bordered? To increase the surface area. Surface area will increase and reabsorption process will occur more, okay. That is why the cells of the PCT are simple cuboidal brush bordered epithelium, okay. So what are the products are reabsorbed? What products are reabsorbed then? Majorly reabsorption process is occurring, no, because simple cuboidal brush border epithelium is present. So what products are reabsorbed? 70 to 80% electrolyte is reabsorbed, okay. 70 to 80% of electrolyte is reabsorbed and water is also reabsorbed and essential nutrients are reabsorbed. You have to mug up it. 3 elements are reabsorbed, that is electrolytes, 70 to 80% electrolytes, and water is reabsorbed and essential nutrients are reabsorbed. This water is reabsorbed, no, that is facultative. Facultative means compulsorily it has to get reabsorbed, okay. No conditional, no condition. Based on condition, the water will not be reabsorbed every single time. In the PCT, facultative, its function is only to reabsorb the water, electrolytes, water and essential nutrients, okay. Now these electrolytes, everything are reabsorbed. To maintain pH and ionic balance, some electrolytes and components will be secreted into the PCT, okay. Selective secretion will occur. Whose secretion? H plus ions, ammonia ions, okay. These two ions will be secreted in PCT and reabsorption of bicarbonate ions also will occur, okay. These are the functions of tubules. Once again, 70 to 80% electrolytes are reabsorbed, water is reabsorbed essential nutrients are reabsorbed. To maintain pH and ionic balance, H plus ion, ammonia compound are secreted into the PCT and bicarbonates are reabsorbed. Okay, this is what you have to remember in PCT. Now let us discuss about loop of Henle. In loop of Henle, see these nephrons are there, no? Nephrons are present in the up to this Bowman's capsule, this PCT, DCT, they are present in the cortex region, right? This loop of Henle is present in the medullary region. In medullary matrix, there will be so much of ions, okay? Ions will be there. On the cortex region, ions will be less. As we go down in the tip of the loop of Henle, ionic concentration will be so high. Here ionic concentration is low. What is the ionic concentration? That is 300 milliosmol per liter. Okay, here ionic concentration is very high, no? What is the ionic concentration? 1200 milliosmol per liter. Okay, you have to remember this thing. I have explained briefly. Now see loop of Henle. You understood, right, what I am trying to say? Loop of Henle is present in the medullary region. In medullary matrix, you will find so many ions are present and some concentration is present. Close to the cortex, ionic concentration is less, that is 300 milliosmol per liter, and near the tip of the loop of Henle there is higher concentration, it is 1200 milliosmol per liter. As we go down from cortex to medulla, the concentration increases. Okay, this is simple. Now the filtrate is there, no? Filtrate is formed in the cortex region only, no? Bowman's capsule, PCT. So its concentration also will be 300 milliosmol per liter. The filtrate which is coming from PCT will also have a concentration 300 milliosmol per liter. Now it should pass down into the loop of Henle, right? Loop of Henle, first descending limb of loop of Henle. Now here the concentration is 300 milliosmol per liter. Now as the concentration is decreasing, no, so what happens as the concentration is decreasing, here the filtrate which is present in loop of Henle also should match with the concentration which is present in the medullary matrix. Here it is 300 milliosmol per liter. Now the filtrate should get concentrated. Water should be removed out so that this 300 milliosmol per liter will get more concentrated, and it will reach to 1200 milliosmol per liter concentration. Okay, that is why the 300 milliosmol per liter filtrate will go down in the descending limb of loop of Henle. Through that process, this descending limb of loop of Henle by nature, it is permeable to only water. Water will be filtered out. Okay, ions will not be filtered because this should be more concentrated. If ions will be removed, still it will become diluted, no? That is why ions are not filtered, not permeable, only water is permeable. And here the filtrate, see, filtrate will be 1200 milliosmol per liter, becomes concentrated. See, becomes concentrated. This filtrate becomes concentrated by descending limb of loop of Henle, permeable to water. Okay, now you see, now it reached to the concentration with the medullary interstitial fluid. The filtrate concentration is same as medullary interstitial fluid. Now it should go upside, right, by ascending limb of loop of Henle. And we know when it is going upside, ascending, the medullary interstitial fluid concentration will decrease. See, it is decreasing, no? When it is going above, upwards, the concentration is decreasing. Now this fluid filtrate concentration also should decrease. Now the filtrate concentration should decrease. So what happens? Water will be, sorry, concentration should decrease. That is why ions are there. No, it is removing ions only, so that concentration will decrease in the filtrate. The ions which are released are NaCl. Okay, so here again filtrate back to 300 milliosmol per liter will happen. Okay, so once again filtrate from PCT will be 300 milliosmol per liter, but in the medullary interstitial fluid the concentration osmolarity will be increasing from cortex to medulla, that is 300 milliosmol per liter to 1200 milliosmol per liter. So the 300 milliosmol per liter will go through the descending limb of loop of Henle. Through that process it should become more concentrated, right? That is why descending limb of loop of Henle is permeable to water, not permeable to ions. So filtrate will reach to 1200 milliosmol per liter, and again it should go back to the filtrate concentration. See, medullary interstitial fluid concentration is 300 milliosmol per liter. It is decreasing again in the ascending limb of loop of Henle. That is why ions only removing. Ions are only permeable to descending limb of loop of Henle, and the concentrated filtrate will come back to normal. Okay, that is 300 milliosmol per liter. And what you this is the mechanism you have to remember, function of loop of Henle. Okay, now reabsorption is minimum in ascending limb of loop of Henle. Here reabsorption process is less, reabsorption is not occurring. Okay, this loop of Henle plays significant role in maintenance of high osmolarity of medullary interstitial fluid. Okay, to maintain the medullary interstitial fluid high concentration, this loop of Henle plays significant role in maintenance. Okay, so we are done with the PCT, we are done with the PCT, we are done with the loop of Henle. Now let us go to DCT. Okay, in DCT what happens? In DCT same as PCT I can say. So what happens? Water is reabsorbed. In DCT conditional water is reabsorbed. In PCT compulsorily water should be reabsorbed, but here in DCT sometimes due to conditions of the body water will be reabsorbed. If you are drinking excessive water, okay, only some specific concentration of water only will be reabsorbed. Okay, and if you are drinking more water, then the remaining water will be gone through to the collecting duct. Okay, it won't be reabsorbed in the DCT. So H2O is reabsorbed conditionally in DCT. Okay, along with that sodium ion is also reabsorbed. See here, electrolytes, water and nutrients are reabsorbed. Here sodium ion means electrolyte only, and water is reabsorbed. Okay, and what are all secreting? Potassium ion, hydrogen ion, potassium ion, ammonia are selective secretion, and bicarbonates are reabsorbed. See, bicarbonate are reabsorbed, hydrogen and ammonia are released here. Here hydrogen, potassium and ammonia are released. What is the function of this? pH and ionic balance is maintained. Okay, sodium and potassium ionic balance is maintained. Sodium and potassium pump will be saying, no. So in that way sodium and potassium ionic balance will be maintained and pH will be maintained. Okay, now let us know about collecting duct. What happens in collecting duct? Collecting duct will produce concentrated urine. It will produce concentrated urine, means the osmolarity again will become concentrated. Okay, how it is maintained? See, now here in collecting duct only so much water will be reabsorbed. Water will be reabsorbed in highest amount so that the urine will be more concentrated. Okay, more concentrated. To produce more concentrated urine, water is reabsorbed and urea is also reabsorbed. Why urea is reabsorbed? See here, urea is reabsorbed in the collecting tubule. Why? Collecting duct. Why? Here the medullary interstitial fluid osmolarity is what? 1200 milliosmol per liter, right? To maintain the osmolarity in the medullary interstitium, urea will be released. To keep the osmolarity of medullary interstitium, that is 1200 milliosmol per liter, urea will be reabsorbed. Okay. Now what are the ions which are secreted? Same, potassium, hydrogen ions are secreted, okay, to maintain the ionic balance, okay. Collecting duct from cortex region to inner medulla, it will be extended, okay. So what are reabsorbed? Water and urea are reabsorbed and potassium and hydrogen are secreted. See, now let us discuss all the functions in this diagram of NCERT. See, recall once again, what did I tell you? Electrolytes are reabsorbed, nutrients are reabsorbed, water is facultatively reabsorbed. And what is the secretion? Ammonia compound and hydrogen ions are secreted and bicarbonate ion is reabsorbed. You might be thinking, ma'am, what is this potassium ion? It should be secreted, no? Why is it reabsorbing? So some amount of water will be reabsorbing, no? Along with that, this ions also will be reabsorbed. Okay, don't think that why is it reabsorbing? It should secrete, no? You don't think like that. Some electrolytes also reabsorbing, no? Along with water, it will be reabsorbed. And potassium also will be secreted. Okay, but just remember, nutrients, electrolytes, water facultative will be reabsorbed and hydrogen ion and ammonium is secreted, bicarbonate is reabsorbed, okay. This is also reabsorbed along with water. This is about proximal convoluted tubule. It is present in the cortex. Now descending limb of loop of Henle. I told no, here the concentration is 300 milliosmol per liter. The filtrate concentration and also medullary concentration. Here the medullary concentration is 1200 milliosmol per liter, no? So that's why it should get concentrated. That is why descending limb of loop of Henle is permeable to water. Okay. Now here 1200 milliosmol per liter. Concentration is there in the medullary fluid and also in the filtrate. Now here it is 300 milliosmol per liter concentration is there in medullary filtrate, but medullary interstitium. This is the concentration and this is the concentration is in inner medullary interstitium, okay. But filtrate concentration is also 1200 milliosmol per liter is the concentration. Now it is going upward in the ascending limb of loop of Henle. Now the concentration of the filtrate should be 300 milliosmol per liter. That's why what happens, it is more concentrated, it should be diluted. That is why ions like NaCl will be released in the ascending limb of loop of Henle. Here it is a thin segment of ascending limb of loop of Henle. This is a thick segment of ascending limb of loop of Henle. In thin segment of ascending limb of loop of Henle, passively ion will be reabsorbed, okay. Here actively reabsorbed. So we are done with the loop of Henle. Now it is distal convoluted tubule. In distal convoluted tubule, recall once again what did I explain. What are reabsorbed? Sodium ion is reabsorbed, water is reabsorbed, no, sodium ion, water is reabsorbed and secretion of potassium and hydrogen. Potassium and hydrogen is secreted and bicarbonate as usual is reabsorbed to maintain the ionic and pH balance, okay. Remember this. And next in the collecting duct, I told you water will be reabsorbing in highest amount for concentrated urine to be filtered out. Okay, and urea also will be reabsorbed in this segment only, because to maintain the interstitial medullary interstitial concentration. Okay, and along with that H plus and K plus ions will be secreted. See here also H plus and K plus ions are secreted in distal convoluted. Same ions will be H plus and K plus will be secreted. Here H plus and ammonia will be secreted. Okay. We are done with the functions of these tubules. Now let us go about mechanism of concentration of filtrate. How filtrate is being concentrated. Okay, so first we will go in this slide and then we'll come back here. Okay, yeah, so already we discussed a little bit in the previous slides. Now let us discuss in detail. Okay, mammals are there. Now we are mammals and we have ability to produce concentrated urine, of course, because 180 liter of filtrate, 180 liter of filtrate is out of 180 liter of filtrate, 1.5 liter of urine is produced, means remaining is reabsorbed and it is concentrated. Okay, so mammals have ability to produce concentrated urine, loop of Henle and vasa recta. See, this loop of Henle and this vasa recta will be helpful in maintaining the production of concentrated urine. See, I explained this in the last slide only. Flow of filtrate between the two limbs of loop of Henle, that is counter current. See, it is descending and it is ascending. It is counter current in opposite direction, no? That is why counter current is there between the flow of filtrate between two limbs of loop of Henle, and flow of blood through limb of vasa recta is also counter current. See, this is vasa recta from peritubular capillary. See, the direction of flow of blood is opposite. This is downward, this is upward. So it is counter current mechanism. And if you see the proximity between the loop of Henle and vasa recta, it is filtrate is going upward and vasa recta blood is flowing downward. It is also counter current. If you check this side also, it is going downward filtrate and blood is going upward. See, counter current. That's what they are saying. Flow of filtrate between the two limbs of loop of Henle is counter current. Between the limbs of vasa recta is also counter current. And between the loop of Henle and vasa recta is also counter current. Okay. We know that from cortex the osmolarity is 300 and medulla it is 1200 milliosmol per liter. Okay. So here proximity between the loop of Henle and vasa recta is counter current. Why? It helps in maintenance of osmolarity 1200 milliosmol per liter towards the inner medullary interstitium. See, this loop of Henle and vasa recta also helps in maintaining the osmolarity of inner medullary interstitium 1200 milliosmol. Okay. Now, how this osmolarity is maintained in the medullary interstitium? Through NaCl and urea. This NaCl and urea will play an important role in creating an osmolarity in the medullary interstitium, and concentrated urine is produced out by NaCl and urea. This is so important. Okay, this is also important. Now we will learn how urea and NaCl is helping in creating the concentration gradient. First we will discuss about NaCl. See students, we discussed in the previous slide, this loop of Henle descending limb is favorable for reabsorption of water because it needs to produce a concentrated filtrate, because in the cortex region 300 milliosmol per liter is there, filtrate is also 300 milliosmol per liter, but in the medullary interstitium 200 milliosmol per liter is there. To maintain that, water is reabsorbed and filtrate will be concentrated. Now here again, near the DCT, tip of the ascending limb of loop of Henle, the concentration of the filtrate should be 300 milliosmol per liter, because the cortex concentration is 300 milliosmol per liter, but it is highly concentrated. Filtrate is highly concentrated. That is why NaCl is permeable through ascending limb of loop of Henle, right? So this NaCl is reabsorbed in the medullary interstitium. This vasa recta is there, no? Vasa recta, which is flowing like this, right? So by the descending limb of vasa recta will receive the NaCl, and it will take NaCl along its tube, blood vessel, and ascending limb of vasa recta will release the NaCl back into the interstitium. Okay, this is the mechanism how it is maintained. And next one is urea. NaCl helps in this way to maintain the medullary concentration. Okay, so medullary concentration is maintained like this. And next one is urea. How urea? We already told that in collecting duct, urea will be reabsorbed, and in the medullary interstitium it is present. But this ascending limb of loop of Henle is there, no? It will carry the urea into the DCT and again into collecting duct. Again urea is reabsorbed. Just this process. Okay, thin segment of ascending limb of loop of Henle takes the urea. This collecting duct is there, no? This urea transport back to interstitium by collecting duct. Okay, this is a counter current mechanism. You have to remember NaCl and urea plays in counter current mechanism in maintaining the medullary interstitium. Okay, just remember this process through which medullary interstitium concentration will be maintained. See, counter current mechanism means what? Maintain concentration gradient in the medullary interstitium. In medullary interstitium, concentration gradient should be maintained. That is called as counter current mechanism. Presence of gradient, easy passage of water from collecting tubule. See, now gradient is present in such a way that water is reabsorbed. Easy passage of the water in the collecting tubule makes the concentrated filtrate. Water is reabsorbed. The filtrate will be more concentrated and urine will be more concentrated. That's what they are saying. Okay. 4 times more concentrated urine is there than initial filtrate. Initial filtrate will be there, no? Than that, the urine which is produced, that is 4 times concentrated. Okay, by this mechanism, counter current mechanism. Okay, 2 types you have to remember: NaCl role and urea role. They may ask questions. So this is the end of mechanism of concentration, concentrated filtrate. How concentrated filtrate is formed. Urea is reabsorbed and this process will continue. Okay, it will be reabsorbed from collecting tubule and received by the ascending limb of loop of Henle, DCT, collecting tubule, again reabsorbed. Little amount, not all. Majorly urea will be excreted, because why? It is a nitrogenous waste. It should be excreted, but little amount of urea to maintain the osmolarity, counter current mechanism. The process will take place like this. And we know sodium NaCl also, it is reabsorbed in the ascending limb of loop of Henle, which is taken by the vasa recta, and again by the ascending limb of vasa recta it will be reabsorbed in the interstitium to maintain the osmolarity. Okay, how it is maintained? See here, it is maintained. Water, it is maintained. You just remember like this. Okay, I think it is clear. Next, regulation of kidney function. How kidney is regulated? Okay, see, for kidney function regulation, 3 hormones will be released. Okay, first hormone is ADH, that means antidiuretic hormone. Diuretic means what? Diuresis, increase fluid increase quantity of urine is called as diuresis. Antidiuretic means decrease the urine content. Okay, for that antidiuretic hormone is released. Next, JGS cells. JGS cells means juxtaglomerular apparatus cells. And next one is ANF. ANF means atrial natriuretic factor. These three hormones are released for regulation of kidney. See, regulated by hormonal feedback. Through hormonal feedback, kidney is regulated. That is by hypothalamus. Hypothalamus, okay. Hypothalamus helps in regulation of the kidney, JGA cells, and certain extent heart also. See what happens. So there is any loss of blood volume. Volume of blood is less. Body fluid volume is less. Body fluid volume is also less, and blood volume is also less, and anic concentration is also changed. Okay, then what happens in the blood vessel? Blood volume is less, body volume is less, and concentration is changed. Then what happens? Osmoreceptors are there, no? They will get activated. When these changes are noticed, osmoreceptors will get activated and it will release antidiuretic hormone or vasopressin. See, diuresis means increased fluidity. Increased urine is passing in larger amount. That's why blood volume is lost, body fluid volume is lost. That is why what happened to prevent the diuresis, increase volume of urine through the excretory pore, we released antidiuretic hormone to prevent the diuresis, increased urine frequency. We release the antidiuretic hormone or vasopressin from neurohypophysis. This is the pituitary, posterior pituitary. From posterior pituitary, antidiuretic hormone or vasopressin is released. Now what it does? It constricts the blood vessels. Blood vessels will get constricted. What happens? Blood pressure will increase, and through blood pressure, renal arteries there, no? Afferent arteriole and then glomerulus. If blood pressure is increasing, what happens? Glomerular blood flow also will increase, and GFR also will increase. Okay, what is another function of vasopressin? Now vasopressin or ADH hormone, antidiuretic hormone, what it does? Antidiuretic means water content, less water content will be excreted in the urine. Diuresis means what? Water amount is more in the urine. Diuresis is occurring. Okay, to prevent this, what we do? We decrease the water content in the urine and make it antidiuretic. Okay, so water is reabsorbed in the later part of the tubule, that is DCT and collecting duct, and prevent the diuresis. Water reabsorption will occur and constrict the blood vessel. Antidiuretic hormone, and blood fluid will increase. Now blood fluid will increase. Okay, switch off the osmoreceptors. Now osmoreceptors should be switched off, because if these are activated, then more ADH will be released, antidiuretic hormone, and again this process will increase. More GFR and more water is reabsorbed, more concentrated urine will be formed. To prevent that, switch off the osmoreceptor when body fluid is increased and suppress the ADH and feedback is complete. Okay, this is the first regulatory hormone, that is antidiuretic hormone or vasopressin from posterior pituitary neurohypophysis. Next one is juxtaglomerular apparatus. We already discussed this, which is formed between the afferent arteriole and DCT cells. Okay, modification of the cells will form juxtaglomerular apparatus, which release the renin. Okay, see, fallen GFR, blood flow is fallen, glomerular blood flow is decreased, glomerular blood pressure is decreased, so GFR is also decreased. What happens? That is why they activate the JG cells. JG cells are activated when these conditions are absorbed, and they release renin. If renin is released, what happens? Angiotensinogen is there, no? Will convert into angiotensin 1 and then further into angiotensin 2. Okay, this angiotensin 2 is a important hormone. Okay, so this will go and activate the blood vessels powerful vasoconstrictor contracts the constrict the blood vessels so much so that increased glomerular blood pressure will occur and blood flow will occur and GFR also will occur. Okay, angiotensin 2 is a powerful vasoconstrictor, and this angiotensin 2 will act on kidney. There is a adrenal gland in the cortex region. They will angiotensin 2 will act and it will release aldosterone. This aldosterone is there now. This aldosterone helps in reabsorption of water and sodium ion from DCT and collecting tubule. From later part of the tubule, water and sodium are reabsorbed. If they are reabsorbed in the blood vessel, so what will happen here? Blood pressure will increase and increase in GFR. Okay, this mechanism is known as renin angiotensin mechanism, JGA apparatus. Okay, now last function is ANF. ANF is atrial natriuretic factor, which is present in the heart. See, now blood flow is increasing. See, here what happens? Blood pressure is increasing everywhere, right? In the blood vessels. If blood flow to the atria of the heart is increasing, blood flow in the atrium, blood flow is increasing, then what happens? ANF is released, atrial natriuretic factor is released. It will cause us blood vessels are constricting, blood flow is increasing, and blood will be flowing more in the atria. That's why ANF will be released in the heart, which will dilate the blood vessels. Now blood pressure will decrease and blood flow to the heart atrium will be reduced. Okay, that's what it is, the function of atrial natriuretic factor. It will be like antagonist to what? Aldosterone and JG apparatus and antidiuretic hormone, because they increase the blood flow by vasoconstriction and all. See, but ANF will be helpful in vasodilation. ANF will act as check on renin angiotensin mechanism. Okay, now let us discuss about micturition. So this is the second last topic of excretion. Okay, so how micturition occurs? Now what is that? Urine through the medullary pyramid, it will be passes through the callus and then a renal pelvis and then ureter, then urinary bladder. It is stored in urinary bladder. Okay, urine is stored in urinary bladder. When this urine is filled in the urinary bladder to the maximum, some stretch receptors are present in the urinary bladder. So these stretch receptors will send signals to CNS, like urine is filled, now it's time for micturition, that is excretion of the urine. Okay, so this CNS, what it will say? It will give motor signals. Actually, it is a voluntary process. Through our control, there is micturition. It is not self-control. Okay, so it will give motor signals, which are voluntary. Okay, motor signals will be given by CNS. Those are voluntary. Now what happens? It has given signals. So the smooth muscles in the urinary bladder will be there. No, they will start contracting, still contracting, so that the urine will be passed out. And here one sphincter will be there. Okay, sphincter in the urethra. So this sphincter will be closing, tight close, so that urine should prevent from excretion. Okay, but now signals are given by CNS for the release of urine. That's why this sphincter will relax and contraction of smooth muscles, so excess fluid urine should pass out. So micturition. Okay, neural mechanism causing it. This is the neural mechanism which is causing it. No, that's why micturition reflex. Okay. So 1 to 1.5 liter per day of urine is passed out. And what is the color of urine? It is light yellow. Okay. And it is a watery fluid, not solid. It is a watery fluid and slightly acidic. That is 6 pH will be there. Okay. Characteristic odor will be there. Okay. Light yellow, watery, slightly acidic, and odor will be there. Characteristic odor. So here you will see how much urea will be excreted. We told that urea, some amount of urea will be reabsorbed from the collecting tubule into the interstitium, right, to maintain the osmolarity. But on an average, majority of urea will be excreted. No, how much? 25 to 30 gram of urea is excreted per day. Remember this. Okay. If in urine glucose is present, then the urine is called as glycosuria. If ketone bodies are present in urine, then ketonuria, urine, glycosuria, ketonuria. Okay. This condition is called as presence of glucose and ketone bodies in urine is indicative for diabetes mellitus. Okay, glucosuria, ketonuria. So urine helps in clinical diagnosis of metabolic disorder. If you give urine for culture and all, we will find the metabolic disorders. So it is of clinical diagnosis, clinical significance. Okay. We have to learn about some other organs in excretion. We know kidney is a major organ for excreting all the nitrogenous waste, majorly urea. Now lungs are there, no? 200 ml per minute of CO2 is removed. Okay. Lungs also act as an excretory organ because it is removing 200 ml per minute of CO2 is removed. Okay, and water is also removed, significant quantity. First organ is lung, which is excretion, and liver also excretes waste products like bile. Bile is there, no? Bile is produced by the liver, which contain bilirubin, biliverdin, cholesterol. Cholesterol is a fat, and degraded steroid hormones. Steroid hormones are also excreted. Vitamins and drugs are also excreted. Liver, okay, it is also excretory organ. Sweat glands are there, no, which are present on the skin. They also release water through sweat. What pasina means? What water is excreted out, right? In water it is little bit salty, right? That is why NaCl is released. Urea is also released through sweat glands, and lactic acid is also released. But what is the primary function of the sweat? For cooling effect only, what happens? Sweat will be released by the sweat glands. In that process, this minute amount of water, NaCl, urea and lactic acid is also released. Okay. Next is sebaceous gland. Sebaceous gland means fat glands. Okay. Fats will be excreted like steroid, hydrocarbon and waxes. Okay. It will excrete these and form a sebum. Okay. Sebum is a protective oily cover for skin. On skin, all these fat contents like steroid, hydrocarbon, waxes will be excreted and they form protective oily cover for skin. Now saliva also excretes some nitrogenous waste. Okay, remember these other organs in excretion, and this is the last topic, that is disorder. In disorder, first thing, urea is the main nitrogenous waste which should be excreted out, right? If urea is in the blood, accumulated in the blood, then it is called uremia. Urea accumulated in the blood, it is called uremia. Why this condition will occur? When there is malfunctioning of kidney. Kidney is not properly functioning. At that time, blood will be accumulated by urea. Okay. So uremia will occur. Uremia is due to kidney failure. Kidney fails because malfunctioning of kidney will occur, and urea should be removed, right? Otherwise, urea in the blood is highly dangerous. That is why urea should be removed. How it is removed? Hemodialysis is a process. So this is the process of hemodialysis, which is so easy. See, now a person will come to hospital, and through the artery of the person, what blood is drained into this dialyzing unit or artificial kidney. Okay, blood will be drained, given into the artificial kidney or dialyzing unit from the artery of a person. Before blood is given into the dialyzing unit, heparin is an anticoagulant. See, in the blood of a human being, anticoagulants are naturally present. That is why the blood will be in fluid form and it won't get coagulated. But this is outside the body, right? It is a dialyzing unit, artificial kidney. The blood will be clot if directly blood is sent into the dialyzing unit. That is why heparin anticoagulant is added and blood is drained into the dialyzing unit. In dialyzing unit, cellophane tube is present, and here dialyzing fluid is present. Okay, dialyzing fluid, it does not have nitrogenous waste. Okay, in this waste is not present, and composition is same as plasma. Plasma composition is present in dialyzing fluid, and nitrogenous waste are absent. And this is cellophane tube. Now, what happens? Now, this blood is there, no? In this blood, nitrogenous waste, urea is present, right? Here, urea is absent in the dialyzing fluid, and here urea is rich, right? From high concentration to low concentration, this urea will pass out into dialyzing fluid and it clears. Okay, blood is cleared from urea, and what happens? It will be sent back to body through vein. Okay, human body through vein, and we add antiheparin. Now we gave anticoagulant to prevent clotting. Now we give antiheparin because in the blood itself there will be heparin, no? So by the returning process we give antiheparin in the vein, and then it is sent back through the vein. So this is the process of hemodialysis. I hope I am so clear. So hemodialysis will occur up to some extent only, but ultimate procedure for treatment of acute renal failure is kidney transplantation from the donor. Donor will be there, no, whose blood group matches with your blood group. So you have to take the kidney of the donor, and kidney transplantation should occur. Hemodialysis up to some extent only we can do, but ultimate method for correction of acute renal failure is kidney transplantation. Functioning kidney transplant, how kidney transplant should be taken from donor. It should be close relative, either sister, brother, uncle, close relative. Okay, to prevent rejection. Okay, organ rejection. Okay, if body rejects the kidney of a donor, to prevent that rejection, it should be taken by the close relative. So first one is uremia disorder. Next is renal calculi. Renal calculi means stone. Stone is an insoluble mass of crystallized salt. Okay, oxalates are there, no? It is a crystallized salt, that is a insoluble mass. Okay, within the kidney. Within the kidney, oxalates will be present, which is a crystallized salt, that is insoluble stone. That is renal calculi, oxalate. Glomerulonephritis means inflammation of glomeruli of kidney. Glomerulus is the tuft of capillaries. Inflammation of glomeruli of kidney leads to glomerulonephritis. So here we come to an end of this chapter. Thank you. I hope I made the concepts clear, the disorders and regulation of kidney and function of tubules, mechanism of urine concentration and urine formation, these types of nephrons and excretory structures, introduction. Hope so I made this chapter very understandable for you. After reading this video, after watching this video, please read the NCERT. NCERT will become very easy if you listen to this video. If you had listened to this video, I think NCERT will become very easy and you can complete it within 15 to 20 minutes. I hope I have reached to you and the concepts are clear.